Jai Hind student, welcome to biology class. So students, in this lecture we will discuss few more important questions of the chapter fundamental units of life. So let's start. Now the first question is, there would be no plant life if chloroplast did not exist. Justify. So you all know chloroplasts, they are the pigments containing chlorophyll. They contain the pigment that is chlorophyll, and this chlorophyll is responsible for the food preparation in plants by the process of photosynthesis. Hence, what will happen if there would be no chloroplast? Then there would have been no plant life, because plants will not able to prepare food. So if plants will not prepare food, they will die. So there will be no sign of plant life on earth. Next, what is cell sap? Give its composition. Liquid content in the vacuoles of plant cell is called cell sap. Now the vacuoles, they contain the liquid content, and that content is known as cell sap. And it contains, that liquid contains sugar, amino acids, proteins, minerals and metabolic waste. Next question, question number 3. What are the functional regions of a cell? So there are three major functional regions of cell, and that is your plasma membrane, or it is also known as cell membrane, nucleus and cytoplasm. These are the three major functional areas or region of the cell. Next, question number 4. Why is Golgi apparatus called the secretory organelle of the cell? Now, so for this you should know the function of Golgi apparatus. So as we all know, the Golgi apparatus, it packages the material synthesized in endoplasmic reticulum, and after the packaging it dispatch it to intracellular and extracellular targets. गोल्गी apparatus का क्या काम है? पैकेजिंग करना, और उसके बाद जब जो भी मटेरियल सिंथेसाइज्ड हुआ कहाँ पे, एंडोप्लाज्मिक रेटिकुलम में, उसकी पैकिंग करना और फिर उसको क्या करना, एक जगह से दूसरी जगह भेजना, जहाँ जहाँ उसका टारगेट है या जहाँ जहाँ उसको इसकी ज़रूरत है. सो दैट इज़ टू इंट्रासेलुलर ऑल्सो, दैट इज़ योर प्लाज्मा मेम्ब्रेन एंड लाइजोसोम्स, एंड टू एक्स्ट्रा सेलुलर टारगेट्स ऑल्सो, दैट इज़ योर सेल सर्फेस. Next question, why do the animal cells do not have cell wall? So animals, they do not have a cell wall because cell wall are incompatible with the way in which an animal moves and grow. So because there is no requirement, basically जो cell wall है, that is made up of cellulose resistant, tough resistant substance होता है, okay, and this helps in providing the protection to the plant, because plants are immotile and they have to face all the adverse environmental conditions, high temperature, strong winds, and still you have seen after that the plant remains still, because this protection is provided by the cell wall. But as compared to plant cell, animal cells जो होते हैं, जो एनिमल्स होते हैं, दे हैव द फ्री लिविंग, दे आर फ्री लिविंग, ओके, दे मूव in search of food, okay, they grow. Now what happens? So the flaccid cell membrane provides the animal cell freedom of mobility and formation of different tissues, which is not present in plant. So animal cell में cell membrane होती है, no cell wall is present, because cell membrane is the flexible membrane, ओके, स्ट्रेचेबल मेम्ब्रेन, फ्लेक्सिबल होती है, जो कि एनिमल्स को फ्रीडम प्रोवाइड करती है, सो दैट दे कैन मूव फ्रीली in their environment in search of food, in search of their friends, everyone, okay. So that's why, and they also have to form different organs for the growth of the body. So that's why it is not present in plant, but it is present in animal. And in plants, apart from this, an extra protective structure is present, that is the cell wall in case of plants. Next, how is endoplasmic reticulum important for membrane biogenesis? Now, membrane biogenesis is the formation of plasma membrane. So, endoplasmic reticulum, they are the main site for synthesis of lipid and protein. यहीं पे synthesis होता है. And the membrane is lipoproteinaceous in nature. That means कि protein and lipids, they are needed for the synthesis of plasma membrane. That's why endoplasmic reticulum perform a major role in membrane biogenesis. What are the functions of nuclear membrane? The nuclear envelope separates the environment of nucleus from the rest of the cell. सबसे पहले तो सेपरेट करता है एनवायरनमेंट को, साइटोप्लाज्म और न्यूक्लियोप्लाज्म को सेपरेट करता है. सेकेंड, इट प्रोटेक्ट्स द जेनेटिक मटीरियल फ्रॉम डैमेज. अंदर, क्योंकि न्यूक्लियस के अंदर ही जेनेटिक मटीरियल होता है, उसको ये प्रोटेक्ट करता है फ्रॉम गेटिंग डैमेज. And the last is it facilitates and regulates exchange of material in and out of the cell. So जो भी मटीरियल्स हैं उनका एक्सचेंज जो है वो जो न्यूक्लियर मेम्ब्रेन में न्यूक्लियर पोर्स प्रेजेंट होते हैं, सो दैट इज़ डन बाय दिस ओनली, क्योंकि मटीरियल्स कीप्स ऑन कमिंग आउट इनसाइड एंड द फॉर्म मटीरियल्स दे आर मूविंग out from the nucleus into the cytoplasm, so that also is done through this nuclear membrane. Now next, in brief state, what happens when? So here are the few options you can see. Dry apricots are left for some time in pure water and later transferred to sugar solution. So क्या होगा, apricots जब हम उसको जो है सबसे पहले जब हम उसको प्योर वाटर में डालेंगे, then it will swell due to osmosis, and when it is transferred to sugar solution, then it will shrink again due to exosmosis, due to the removal of water from the cell. Next आते हैं, a red blood cell is kept in concentrated saline solution. So again the RBCs will shrink due to exosmosis because the solution is concentrated. Next, the plasma membrane of the cell breaks down. So what will happen, though, if the plasma membrane will break down, then what will happen, it will lead to scattering of cell organelles and there will be no functioning of the organs, because plasma membrane के अंदर ही प्लाज्मा मेम्ब्रेन एक कवरिंग की तरह होती है एंड ऑर्गेनल्स आर प्रेजेंट इनसाइड, तो सब स्कैटर्ड हो जाएंगे. ओके, नेक्स्ट, Rio leaves are boiled in water first and then a drop of sugar syrup is put on it. So what will happen? There will be no change because during the boiling करते ही समय जो cells जो हैं वो dead हो जाते हैं. So no endosmosis, no exosmosis. Last question is Golgi apparatus is removed from the cell. So Golgi apparatus का function क्या है? That is packaging, storing and transfer of proteins. So these all functions will be affected if the Golgi apparatus is removed from the cell. So these are few questions. I hope you all have understood. Thank you everyone.